let's continue on. You will remember in the last lecture we ended up talking about um, DNA and chromosomes, and we had mentioned the term histone, and so I wanted to go into that a little bit more detail. A histone is a protein that acts like a spool which winds up certain parts of DNA. So on this diagram, you see we have a DNA strand, and this DNA strand is wrapping itself around this part right here. And this is a protein, and when this wraps around enough, it begins to associate with other wrappings and other proteins. And when you get DNA wrapped around a histone forming series of beads like this, that's what's known as a nucleosome. And as it shows here, there's actually the spool or central histone that wraps the DNA, and then there's a spacer, and then another histone or central histone, and there's a connection in the DNA between those two, and this is called a spacer histone here and a central histone here. But this piece right here, when you get several of these together, it's called a nucleosome. Now, once you get multiple nucleosomes together, they form, and you have a series of aggregations of larger and larger, more and more compacting of the genetic material until you end up with what's known as a condensed chromosome, which is shown on this point right here. And that's the smallest package of DNA that can exist inside the nucleus. And so this is as compact and tightly wound as it can possibly be done. And you can see here, you take a little section of this and it forms this big piece right here, and a little section of this to this piece, and a little section of this to this piece, and a little section here, and you have the nucleosome. And in each case, it's getting littler and littler. And so this is an enormously condensed piece or aggregation of DNA. Now, we're going to move on and begin to talk about a process known as mitosis. And mitosis is important. It's the asexual reproduction that occurs in eukaryotic cells. And I want you to spend a little time making sure you understand the various stages in mitosis, and we'll go through that very carefully. And then probably in the next part, we will talk about something called meiosis, which is unique to the sexual cells in the organism. So let's take a look at mitosis. First of all, most cells are not dividing. And so most cells are between mitotic events. And that phase or time interval between cellular reproductive events is called the interphase. And that's what this is supposed to show, is a cell that's actually right before it begins to go into mitosis. And the reason I say that is you can see here you have the centrosomes here, and then the aster, which we're going to mention those in the near future. But what begins to happen during interphase is that chromosomes begin to shorten and begin to become more distinct and easier to see. You can also see here's the nuclear membrane, the nucleolus, which is where RNA is produced, and the plasma membrane here. So once you have an interphase like this and one mitotic event, the total time between that includes both an interphase interval and a mitotic event is called the cell cycle. So when a individual or teacher or someone that you're talking to mentions the cell cycle, that's what they mean. They mean one interphase event and one mitotic event. And the cell that eventually undergoes mitosis, like this one shown here, is called the mother cell. Now, the mother cell doesn't really become the mother cell until it replicates or duplicates its DNA to form two sets of DNA and also form centrioles. So when these centrioles become visible and easy to see and the DNA is replicated, then it becomes what's known as a mother cell. So the first stage of the mitotic event is called prophase. And prophase is a point where the duplicated chromosomes, which have already occurred, they begin to become more condensed and again more easy to see. Then after they become condensed, they attach to something called a centromere. And at the same time that the centromere happens, and the centromere is this central portion. So here you have a duplicated chromosome right here. There's one on this side, one on this side, and in the center here is the centromere. Now, they're not always in the very center, okay? Sometimes there's the centromeres on one end or the other. But in this case, so you can see it, they put them in the center of all these duplicated chromosomes. So these are two chromosomes that have been duplicated, and here's another one that's been duplicated. And at the same time during prophase, the centriole or centrosome begins to migrate toward the poles of the cell. And so the centromere is as I said, the region that joins two chromatids that have been duplicated. And those chromatids that have been duplicated are called sister chromatids. So this is the later part of the prophase. And during the later part, you have something called the aster, which is the extension of microtubules from the centriole in all directions. It's actually probably better seen on this diagram. You can see this is the aster right here as it comes out from the centriole. Now these centrioles haven't actually moved to the poles. One of them has, but the other one needs to come down here. But the aster are the microtubules beginning to come out. So let's go back to this diagram. And as the aster begins to extend, the microtubules begin to extend out from each of the ends of the cell. And these spindles continue to move out. They're called spindle fibers. And these grow out from the centrioles and they extend until they attach to the centromere. And when they attach to the centromere, they're called the mitotic spindle. So you have the chromosomes lining up along the equator, or what's called the equatorial plane here. The microtubules moving out and attaching to the centromere. And then at this point, you've entered what's known as the metaphase. And so here's metaphase over here on the left. The chromosomes are lined up, and you can see the mitotic spindle is formed, and they're attached all along this equatorial plane here. And then at the next step, the chromosomes are pulled apart, and they're pulled by their centromere, so that's why they have this shape that looks like this, and like this. And you have basically separating, so you can see that they're going to form two nuclei. And this stage over on this side, when they're separating like this, is called anaphase. This is the point in which the chromosomes are actually separated and begin to migrate to the opposite poles of the cell. And then the last phase is shown here. The last phase is telophase. And at this point, the spindle begins to break down. You can't really see it as clearly. You see a little bit of something there, but it begins to break down. And then what's characteristic of the telophase is this formation or pinching in of the plasma membrane. It begins to constrict here until ultimately you end up with two cells, and those two cells are called daughter cells. And you have one nucleus here, one nucleus over here. So those are the 4 stages. Sometimes they talk about 5 stages. The stage they're including when they talk about 5 stages is they're including interphase as a stage. So you have interphase, and during interphase is when the DNA is actually replicated. Then you have prophase, followed by metaphase, anaphase, and telophase. And so as I said before, you'll probably want to go through this part a few times and there's some really nice pictures in your book that you can go through that explain this in detail. Now, in order to understand the next part, we have to understand chromosome typing and how chromosomes are paired together. The human has 46 chromosomes, and the 46 chromosomes come in pairs, okay? And those pairs are called homologous chromosome pairs. There are 2 chromosomes, and here's an example here. This is chromosome 1. There are 2 of them, and they're very similar. Now, they're not identical. They're similar. And because they're similar but not identical, they're called homologous as opposed to some other term. And so the chromosome pairs are similar to each other, and they're extremely different from other pairs, okay? And you'll notice that they're about the same length and they have the similar banding here. But as I said, they're not identical. Now, a term for each one of these 2 members of a pair is a homologue. So you have one member here, one homologue, and a second homologue. Those 2 together make up a homologous chromosomal pair. Now, the sex chromosomes, now this is a female, so it's actually hard to show the difference here. The 23rd chromosome, which is down here, is also known as the sex chromosome. And these determine the sex of the animal, whether it's going to be male and female. And in females, they are also very similar, very homologous. But in males, if we had a male up here, it would be very easy to tell the difference because there would be one large one that looks very much like this one, and that's called the X chromosome. And then there's a little dinky one that looks like this one over here, and that's called the Y chromosome. And that's the way a male looks. That's the way a male pair looks. Now, what I've been pointing out here, by the way, is something called a karyotype. A karyotype is when they take a cell that is in metaphase and they freeze it in metaphase, and they stain that frozen cell. When I say freeze it, I mean they stop it from changing or stop it from growing. They don't actually put it in a cold place and freeze it literally. And then what they do is they stain it with a special stain, and then they take a picture of that, and then they basically cut out all the chromosomes that are there, and they pair them up like this based on their length and their banding. And then that whole process of making a figure of the chromosomes captured and arranged in pairs this way is called a karyotype. So this picture right here is called a karyotype. Now, because this cell had its chromosomes in homologous pairs, it's called a diploid cell. And a diploid cell is a cell that has all of its chromosomes in homologous pairs. Now, all the cells of the body, with 1 or 2 exceptions, are diploid cells. The most common exception is the sex cells. That is, the cells that are responsible for the production of a new organism. Sex cells are also called gametes. They're also called sperm and egg. And when a cell has only one of the 2 chromosomes in a homologous pair, so when it only has one of each of these, one of these, one of these, and one of these, etc., so it has 23 instead of 23 pairs, then it's called a haploid cell. A haploid cell is a cell that has exactly half as many chromosomes as a diploid cell. It has half as many chromosomes as a normal cell, and it's normal if its function is to be a sex cell. It is not normal if the cell's function is for something else. And in our next part of the lecture, in the last part, we're going to talk about how we produce haploid cells. We talked about how we reproduce diploid cells, and that's what was called mitosis. So now we're going to move on to, and the next part we're going to talk about something called meiosis, which is a process that results in haploid cells. And then we're going to finish up by talking about how viruses work genetically.